Welcome to Bio class. Today we are going to discuss one of the important part, that is DNA replication. DNA replication. Okay, replication, it means that synthesis of the DNA. It is a unique property of the DNA that it controls its self duplication, and the process of the self duplication, this is known as replication. It's simple, it is making a copy. So one DNA strand, it turns into two copy of it. Okay, in eukaryotic occurs in nucleus during the interphase S phase, synthesis phase of the cell cycle. But in prokaryotic, it is occurs in cytoplasm, and it is almost a continuous process. Now, several fundamental characteristics are there that we will discuss. Now, come here, and in here it is semi-conservative process. The scheme, of its division to semi-conservative. You have to understand this one. What is semi-conservative process? If we take one example, suppose here is a pair of gold ring. All right, you can see that pair of gold ring. You can just the DNA strand, you just compare it to the DNA strand of it. Now, if we want to make it copy, so what we will do? Normally, we will give one goldsmith. So that goldsmith will make that copy. So by looking this one, will make that copy. So this method, it's known as conservative method. But if we will not do that, if we will do some other thing, suppose if I do that this one, if I give it goldsmith A and another one of the pair, if I give the goldsmith B, it means that I am giving to other. Goldsmith A, by looking this design, he will make another one here, and goldsmith B also looking the pair. Here also we can make the exact copy of it, but this method is called semi-conservative method. Okay, so DNA is replicating, DNA is making its copy in semi-conservative method. Okay. Okay, to prove that the DNA is a semi-conservative type of replication, Messelson and Stahl did the one experiment, that very famous experiment, Matthew Messelson and Franklin Stahl performed the experiment in 1958. What they did, they took Escherichia coli, and they grew it in medium ammonium chloride, which is having heavy nitrogen, 15, normally it is 14, I think you know, 15 nitrogen, heavy isotope of nitrogen, and for the many generation, you know that the nitrogen is the one part of the DNA. So nitrogen, in 15 was incorporated here in the synthesized strand. Okay, it means that it will be the in strand that nitrogen will be there. After that, all these things, it is centrifugation by the cesium chloride. Cesium chloride, it shows the density gradient. If we see this one, this cesium chloride having the property that itself, it is, if it is more dense, then it is found in the bottom. If it's less, then above. In that way, it's can make the layer. Okay. So here we are talking about the heavy and the light type of nitrogen, and show that here also that it can form the layer when we are using that heavy nitrogen, it is found in the bottom layer. And here note that this nitrogen, we are actually it is radioactive, but we are not treating it as a radioactive. Here only the based out on density, that is separated. Okay. Now next part, we are transferring this heavy nitrogen in some other normal medium. Okay. So you have to see that your Book's diagram at first. Now in normal medium, ammonium chloride, that is 14 it is there, and here also the multiplication is there, and we will observe that what type of strand we are getting in that. If we take help with this video clipping here, the first you see this one, this is heavy nitrogen, and now it is transferred into normal. Okay, it is transferred into normal. Let's see this one. Here equally it doubles every 20 minutes. So every 20 minutes we are getting the different result. Okay, look at here. If we can return this one. Look at here. Every 20 minutes when it is so what we have to do now, we have to take DNA from every 20 minutes, because every 20 minutes it will divide, equally will divide, so the DNA will divide, and will isolate the DNA, and after that we are keeping inside the cesium chloride, that gradient centrifugation, and see the layer. Let's take a look at the next part of it. See that the first when it is getting, that is the heavy nitrogen. Then second, after 20 minutes we are getting the layer above which is intermediate. It is not heavy. It indicates that now it is the 15 and the 14 both the strands are there, because it is now we are in 14. Now see that the next layer we are getting when it is in 14, because in 14 layer now increasing, because it is in 14 layer. Now it is 50%. Look at here. If we keep it 60 minutes, so that this layer will increase. So from that video, that it is clear that the first, when it is the in heavy nitrogen, that it shows the layer. So both the strands, it was in heavy, and it shows in the red color here. You can see that this one and this and this. Okay. Next, when we are keeping in normal, so that here it is one intermediate band appears, intermediate. This indicates that it is semi-conservative. Look at here, the intermediate band. It means that one is heavy, another is light. Heavy and light, both it is there. If we increase it in 40 minutes and 60 minutes, so that the increase of that normal, in 14 it is increased. So 50, after that 75, 25. In that way it is increasing. It means that from the one strand, another strand will form. So this is the semi-conservative, the way of DNA replications. Okay. So next part we can uh, go that is the masonry of the enzyme. I'm making that the second part of this video. Okay. So second part of the DNA replication it will be discussed.